Good morning. It's March 28th, and this is your daily brief in science. Here's everything you need to know. A new study spanning 22 years sheds light on the movement patterns of elephants in northern Kenya. Tracking 157 elephants from 1998 to 2020 using GPS data, researchers have uncovered critical insights into how these animals navigate their environment. Utilizing an innovative modeling method known as Enerscape, the study estimated energy costs based on factors like body mass and terrain slope. By integrating satellite data on vegetation and water availability, the research provides a comprehensive view of elephant movement dynamics. The findings have significant implications for wildlife conservation, particularly in designing protected areas and migration corridors. Lead researcher Dr. Emilio Berti emphasized the study's importance for conservation planning, especially in establishing dispersal corridors and understanding the energy costs associated with movement. Notably, the study revealed that 94% of elephants avoid steep slopes and rough terrain, indicating a strong preference for energy-efficient travel. Furthermore, 93% of elephants tend to move toward areas with higher vegetation productivity, seeking resource-rich environments. While water sources play a crucial role in influencing elephant movement, individual responses vary. Some elephants stay close to water, while others travel farther, highlighting the complexity of their decision-making processes. This research underscores the need for conservation strategies that consider individual habitat preferences, particularly concerning access to water. To analyze how elephants choose their paths, researchers employed statistical methods, including step selection functions. These methods compared visited locations with nearby alternatives to identify key environmental factors influencing movement. The results also offer insights into how elephant movements may adapt to climate change, potentially affecting energy costs and resource availability. Given that African elephants require around 330 pounds of vegetation daily for survival, their ability to efficiently find food is vital for their well-being. Future research will aim to refine energy landscape models by incorporating seasonal changes, human impacts, and climate change effects on elephant movements. The rise of Candida auris, a deadly drug-resistant fungus, is raising alarms across the United States. In 2023 alone, over 4,500 infections have been reported, a staggering increase from just 51 cases in 2016. The Centers for Disease Control and Prevention, or CDC, has issued urgent warnings about C. auris, highlighting that no drug currently exists to effectively eliminate it, making it a critical threat in healthcare settings. C. auris infections are particularly dangerous, with high mortality rates among hospital patients who have pre-existing health conditions. Unlike airborne illnesses, this fungus primarily spreads in healthcare environments through contaminated medical equipment and surfaces. It can survive on various surfaces, including countertops and doorknobs, and is often transmitted via medical devices such as catheters and breathing tubes. Patients at the highest risk include those with serious underlying health conditions and individuals using invasive medical devices, such as those who are intubated. In response to the increasing threat, hospitals in California have implemented screening procedures for patients coming from high-risk settings to isolate those infected. However, infection control is complicated by the fact that many EPA-registered disinfectants commonly used in hospitals are ineffective against C. auris. The CDC emphasizes the need for improved surveillance, enhanced laboratory capacity, rapid diagnostic tests, and strict infection control measures to manage the spread of this fungus. Ongoing research is focused on developing new treatments for C. auris as part of a broader effort to address antibiotic resistance globally. Recent reports indicate a surge in C. auris cases in Georgia and concerning rates in Florida hospitals, underscoring the ongoing public health threat posed by this fungus. While C. auris does not typically threaten healthy individuals, it remains a significant risk for patients in healthcare facilities, particularly those who are already ill. Researchers at the University of Leeds have unveiled a revolutionary tiny robot known as the Oloid Magnetic Endoscope, or OME, designed to detect and treat bowel cancer without invasive procedures. This innovative device, roughly the size of a penny, is equipped with a small ultrasound sensor that captures high-resolution 3D scans of the gut walls, providing a compelling alternative to traditional biopsy methods. The OME utilizes a 28 MHz micro-ultrasound array to generate detailed 3D images that replicate those obtained from standard biopsies, enhancing diagnostic capabilities significantly. This technology allows for what are termed virtual biopsies, offering immediate diagnostic data without the lengthy wait times associated with physical biopsies. Professor Pietro Valdastri noted that the OME facilitates in situ tissue analysis and immediate diagnosis of colorectal cancer, contrasting sharply with traditional methods that can take weeks for results. The research team pointed out that this device improves patient comfort, reduces the need for repeat procedures, and alleviates the anxiety linked to waiting for cancer results. Early detection of bowel cancer is critical for successful treatment outcomes, and the OME could greatly enhance the chances of identifying the disease at an earlier stage. The design of the OME leverages advanced materials and engineering to create a minimally invasive solution that may lessen the need for traditional surgical procedures. Its unique oloid shape enhances mobility within the gastrointestinal tract, allowing for safer and more efficient scanning procedures. Testing has confirmed the OME's efficacy in simulated human intestines and pig intestines, with human trials expected to commence in 2026. This research represents a collaborative effort among leading scientists from various institutions, highlighting an interdisciplinary approach to advancing medical technology. The study detailing the OME was published in Science Robotics, marking a significant milestone in the field of cancer diagnosis technology. Researchers at the University of Chicago have unveiled a groundbreaking imaging technique called volumetric DNA microscopy. This method enables the creation of detailed 3D maps of genetic material in living organisms, allowing scientists to visualize the spatial locations and interactions of individual genes. Traditional genetic sequencing methods often fall short, but this innovative approach overcomes those limitations. Volumetric DNA microscopy utilizes unique molecular identifiers, or UMIs, to tag DNA and RNA molecules. This tagging facilitates the construction of a molecular network that encodes the positions of these genetic materials. Joshua Weinstein, Ph.D., who has led the development of this technology, compares the mapping process to cell phones pinging each other to determine their locations, highlighting how molecular interactions can reveal genetic proximities. On March 27, 2025, a study published in Nature Biotechnology showcased the first application of this method, successfully creating a complete 3D DNA map of a zebrafish embryo. The implications of this technology are significant, with potential applications in enhancing immunotherapy and developing personalized vaccines by providing comprehensive insights into genetic expression in complex tissues. Unlike traditional microscopy, volumetric DNA microscopy does not require prior knowledge of the genome, making it versatile across various biological contexts, including tumor environments and immune responses. The study received additional funding from the Damon Runyon Foundation and the Moore Foundation, as well as support from the National Institutes of Health and the National Science Foundation. Weinstein and postdoctoral scholar Nianchao Qian co-authored the study detailing this pioneering application, which could revolutionize our understanding of genetic expression in numerous contexts. Researchers from the German Primate Center and the Max Planck Institute have made a groundbreaking discovery, identifying two unique genes that evolve exclusively in humans and play a significant role in the development of the human cerebrum. These genes work together to enhance the multiplication of brain progenitor cells and facilitate their transformation into nerve cell precursors, which are essential for the evolution of our brain's size and complexity. One of the genes boosts the proliferation of brain progenitor cells, while the other aids in converting these cells into neuron-producing progenitor cells. This study sheds light on the evolutionary history of large brains and may help in understanding developmental disorders and neurological diseases. Necile Esiok, the study's first author, highlighted that this research enhances our understanding of brain development and could lead to new therapeutic strategies for brain malformations. Lead researcher Michael Haida noted that the study's results validate both animal and alternative methods, potentially reducing the need for animal testing in future research. The research combined various methods, including experiments with mice and chimpanzee brain organoids, demonstrating the effectiveness of alternative research approaches alongside traditional ones. Published on March 26, 2025 in the journal Science Advances, the study is titled A Dyad of Human-Specific NBPF14 and NOTCH2 NLB Orchestrates Cortical Progenitor Abundance Crucial for Human Neocortex Expansion. The German Primate Center focuses on biological and biomedical research involving primates, contributing to fields such as neuroscience and infection research. This coordinated gene interaction is believed to be pivotal in the evolutionary expansion of the human brain's size and complexity, offering insights into the origins of developmental brain disorders and neurological diseases. Haida emphasized that the complementary results from different methods reinforce the study's significance and may reduce future reliance on animal testing. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. We'll be back Monday with everything you need to know.